blue session. We have 4 talks. The first one is by Vincent Ragusa and Clifford Foam, Augmenting Evolution with Bio-Inspired Super Explorers. Vincent, you're ready to share your screen?
Yes, can everybody hear me? Excellent. All right. Well, we're all here because we have a shared interest in artificial life. And one of the things that life famously does is evolve by natural selection. So today I'd like to talk about how artificial evolution can be augmented by a technique that my collaborator Cliff Bohm and I have developed called super explorers. So these diagrams here are simplified fitness functions or fitness landscapes, and fitness increases along the vertical axis, while the horizontal axis represents similar genotypes. On the left is a depiction of directional selection, the means by which evolution gains fitness on short time scales. Mutations occur during reproduction, and that creates a distribution of genotypes. And when mutations and selection act in the same direction, the population migrates across the fitness landscape. When a population is at a local optimum in the fitness landscape, selection acts from essentially all sides, and the inflow of mutation and selection against low fitness individuals is balanced, and it keeps the population stable on the optimum. However, stabilizing selection can be an obstacle to future improvements if the regions of higher fitness are on the other side of a low fitness valley. So in general, the process by which a population that is stable on a local optimum is able to escape and discover higher fitness, we call valley crossing. And there are a number of variables that affect how easy or difficult valley crossing is. The opposing forces of selection and mutation give us 2 ways to help cross valleys. We could decrease the selection strength, or we could increase the mutation rate. Both of these choices spread the population thinner across the fitness landscape and can, if taken too far, prevent the population from detecting and stabilizing on fitness optimum. So there's another way to make valley crossing easier, which is to have more organisms in the population. But this, in a laboratory setting or in computer simulations, is often not a good option because of the increased demand for resources that a larger population incurs. So in this work, we're going to focus primarily on reducing selection strength, but we're going to try and avoid those pitfalls of losing detectable fitness gradients. So when a population experiences a range expansion, a subset of the organisms in the population experience reduced selection, while the rest of the population experiences normal selection. So to see how this works, let's imagine we put a population comprised entirely of this green genotype in an empty petri dish. And then let's follow what happens to the descendants of some of the organisms. So when the green organisms on the outside edge of the population reproduce, some of them may mutate to the yellow genotype. And the yellow genotype, now on the outside edge of the population, when it reproduces, some of their offspring may mutate to the orange genotype. But at the same time, the yellow genotype that are not on the outside edge, they're forced to compete with the green type and they're defeated. In the same fashion, some of the orange on the outside edge may mutate to the light red, while the others are defeated by green. And some of the light red may mutate to dark red, while the remainder are defeated by green. And finally, in this scenario, some of the dark red may mutate to a blue genotype, which in this case is a higher fitness than the original green type. And from here, the blue type would go on to outcompete all of the other types and fix in the population. And this is how range expansion can reduce selection almost to neutral drift. The organisms on the outside edge, they don't compete with anyone, and so they can accumulate deleterious mutations without any penalty. Importantly, though, if the blue type had not been discovered during this drift, the green type would not have been lost. And this is not the case when you simply reduce selection on the entire population at once. So by allowing only a subset of the population to drift, the range expansion has the benefits of reduced selection without the negative aspects of losing good fitness. And so what we wanted to do is we wanted to make use of this range expansion benefit for valley crossing. And we wanted to do this without simulating geographic territory. So the way we do this is we simulate the reduction in selection strength along the leading edge of range expansion by allowing some organisms to undergo random walks. We call these special organisms super explorers, and they produce one mutated offspring every generation, regardless of their fitness. Eventually, the super explorer lineage is terminated, and when this happens, we say that the super explorer has decayed. Decay happens randomly with a predetermined probability, and the probability controls the average length of the random walk, and so therefore controls how far from the starting point the super explorer could potentially travel. The collection of all super explorers we call the super explorer pool, or the SEP, and the number of explorers in the pool controls the intensity of the search that the super explorers do. In order to maintain a fixed pool size, when a super explorer decays, we replace it with another organism. And the method by which we replace is a free choice. In this work, we compare 3 different replacement methods. The 3 methods that we chose are each based on the fitness of the organism that we're going to use for the replacement. And this choice was motivated by the desire to restrain the super explorers to areas near high fitness. The first method is to randomly select a replacement from the population, fitness proportionally. And some of you will note that this is the typical selection method used for digital evolution. The second method is to replace from the most fit individual in the current population. So we make a copy of the best organism. And the last method is to replace with the most fit individual that has ever been recorded during the entire runtime of the simulation. We call that the global best. So in quick summary, there are sort of 3 parameters for the super explorers. There's the pool size, which controls the intensity of the search. There's the decay rate, which controls the range of the search. And there's the replacement method, which determines the starting point of each of the lineages. And we'll be varying all of these parameters to kind of see what they do. Now, before we look at that, since super explorers are meant to replicate the dynamics on the leading edge of a range expansion, we also want to make sure that we keep the dynamics expected in the bulk of the population. And so this, we employ a standard digital evolution algorithm that proceeds by selection and mutation. And we add the super explorers into the system. We just connect the 2 together by the interactions between the organisms. So the standard evolutionary algorithm, which we call the active selection pool, or the ASP, it is allowed to select from the super explorer pool. And this allows the main algorithm to leverage any discoveries made by the super explorers. And at the same time, the replacements for decayed super explorers can be chosen from the ASP, ensuring that the starting points are located somewhere near the population. And lastly, in order to use the global best replacement method, we also have this max pool, which simply stores a copy of the best thing that we've seen so far. So now what I'm about to show you are the outcomes of a bunch of evolutionary simulations that use the super explorer technique. This data was recorded from experiments on an NK fitness landscape. The NK fitness landscape can be tuned to a desirable degree of deceptiveness. So in other words, we can control how difficult the fitness valleys are in the fitness landscape, how hard they are to cross. And here we've chosen a moderate difficulty. We run the evolutionary simulations on the NK landscape, and we vary the SEP size, the decay rate, and the replacement method. So in a minute, I'm going to reveal a heat map of the final fitnesses of each run. The outcomes are going to be arranged so that the decay rate increases along the x-axis, and the SEP size increases from top to bottom. So first, we'll look at the results from the global best replacement method. So first note that the red row at the top represents the fitness of the control experiment. And this is true in the rest of the figures as well. So this is the outcome of evolution when there is no super explorers to provide additional benefits. And we see here that nearly all of the configurations of the super explorers provide some benefit over the control. Most notably, large super explorer pools with very low decay rates perform the best with this replacement method because there are a large number of super explorers and they can drift for a really long time. And so they can explore vast expanses of the fitness landscape. Keeping a copy of the global best here provides the benefit that discoveries cannot be lost. And that's critical for super explorers in this region of the setting space. Next, we'll look at the population best replacement method. So we only can take the best of the current population. And you'll notice that there is very little difference between this and the previous replacement method. This is because the replacement method is still able to maintain a high enough frequency of good individuals in the population, such that there are still some good ones around when replacement is needed. And so we're still making sure that we don't lose good individuals in the population. But finally, we're going to look at the random fitness proportional replacement method. And here we see a strong contrast with the first two. So in particular, many configurations of the super explorers perform similar to the control. And in particular, it's those with high decay rates and few super explorers. But despite this, we still do see improvements over the control in this yellow band here. And that tells us that, the additional exploration power of the super explorers is responsible for an increased rate of valley crossing. Interestingly, the region of lowest decay rates and largest pool sizes, they perform nearly 7% worse than the control. And this highlights the importance of keeping good solutions when they're discovered instead of ignoring them in favor of more exploration. This happens implicitly in the first two replacement methods, but not in the third one. Also in each plot, we've marked specific settings with either a star or an X. These correspond to the settings with the best improvement over the control and the worst decline over the control, respectively. And this table here summarizes the statistical significance of those changes. And you'll note that all of them are statistically significant, except for the decreases in the first two methods, which are practically the same as the control. Before I go on, I'd like to say that we do run many more experiments on other fitness landscapes, and those results offer additional insights into the behavior of super explorers and evolution in general. But I won't be showing those results here for time. So if you're interested, I really do encourage you to go read the paper where all of that is discussed in detail. So I want to talk about some interesting future directions. There are many that this could go in, but I'll just highlight some that I find the most interesting. So first, our technique for augmenting evolution is novel as far as we know. It's distinct from other similar sort of multi-population models like island models, for example. And it's different in that instead of keeping the organisms separate, we are keeping the exploration models separate, or the selection methods separate. And so as a result, the super explorer pool is not the only search or optimization model that could be used. Consider particle swarm optimization, for example, which is another bio-inspired search technique. It could easily be used to augment evolution in the same way. But it doesn't need to replace the super explorers either. You could simply add it to the ensemble. In fact, you could add as many different selection or search algorithms to the ensemble as you like. Also, the global max pool, there's no need for it to do something simple. We could replace it with something much more sophisticated, like, for example, the map elites storage paradigm. The possibilities here are basically as numerous as the number of algorithms you could choose from. And that's basically it. So thank you all for giving me your time. I'd like to thank one more time my collaborator, Cliff Bohm, and I will gladly take any questions you have for me.
Great, thank you, Vincent. Thank you also for being spot on time. If you want to ask a question, please raise your hands and the participants and I call you out to so that you can ask your question directly. If you don't have a microphone, you can also just put it in the chat and I can read it out. So see, there is a question from Seth Bullock. How does a random exploration approach cope with a much higher dimensional search space?
So it's a good question. Our experiments so far have not tested high dimensional search spaces. So it's an interesting question because random walks are inherently sort of wasteful. One of the things that we would like to do is to sort of replace the random walks with a more sort of a more directed evolution model or more of a directed mutation model, so that we get more of a radiation from the starting point instead of just complete random walk. And I think that will sort of address the scalability of the dimensions in the fitness landscape.
Great. And another question in the chat, Mia Katrin asks whether when you introduce the super spreaders, the super explorers, whether you reduce the population size or whether you have more samples per generation than in the control.
So we did control for the population size. So the population size here is fixed at 1024. And so the number of super explorers along the y-axis here is actually showing more or less the percentage of super explorers. So it's not, we didn't just add individuals to the population, which would have inherently boosted exploration. We made a trade-off with the other organisms so that we could control for that variable.
Great. Maybe I also contribute one question. It's an interesting concept to try to keep these super explorers alive. And during your talk, it reminded me of kinship, so that the fitness of one individual does not only depend on its personal fitness, but of the fitness of the whole population. You see these two concepts related? Are they different? So alternative explanations, or is maybe kinship and implementation for these super explorers?
So kinship in terms of fitness sharing, I don't think is very closely related, but it is important that the super explorers are a continued lineage, as opposed to the way drift normally happens in a population, which is usually just by the sheer random probability of drifting, according to the standard sort of selection paradigm. So the fact that the lineages are continued, mutations, that means that multiple deleterious mutations can accumulate, and that's really how we get valley crossing. It's the large sum of mutations that gets you, across fitness valleys. And so the relatedness in terms of sort of parent to child lineages, that's what's important here.
I see, thank you. I don't see any other question. So we go right on schedule with the next speaker, which is Jacob Ashworth, Lyra Lee and Jackson Shen, Edward Kim, Zach Decker, and Jason Yoder on the evolution of developmental strategies in NK fitness landscapes. And as I understand, this is a video presentation.
Ladies and gentlemen, I am Edward Kim, and we are a research team from Rose-Hulman Institute of Technology. Today, we're very excited to present our work on the evolution of developmental strategies in NK fitness landscapes. To contextualize our work, we look at how nature produces adaptive organisms. First, there's learning. a well-known source of adaptability that has also been extensively emulated through processes such as machine learning. Then, across multiple generations, there is evolution. What is often overlooked, however, is development. Development is distinct from learning in that it involves structural changes in an organism with varying degrees of plasticity at different life periods, such as in sensitive periods. Development is also uniquely advantageous for artificial life, such as in its scalability with simple representations from which significant complexity can emerge. In the triangle, the relationship between learning and evolution has been frequently explored. However, there has been little progress in examining the relationship between development and evolution, which has become the motivation and objective of our work.
We reviewed 3 areas of relevant literature and their shortcomings. The Evo-Devo models have been mostly based on Evo-Devo model organisms that has led to severe challenges and technical difficulties. Since these models are based on empirical optimizations, which are largely species-specific, there is a need of a simplified and a general model. One of the early models created from Hinton and Nolan, they explored relationship between learning and evolution. Another abstract model, NK model, is a viable candidate that captures high-level processes in multiple fields. The developmental models have a complicated inferential nature, which they are highly specific, such that we identified a gap of abstract developmental models. Given that most previous models are complex, lack significant mechanisms, and lack tasks with epistatic interactions, we formulated a novel way of modeling developmental processes.
Within our model, we have 3 main areas. First, the NK fitness landscape, which is a fitness landscape with tunable size and ruggedness. Next, we have the developmental strategy, which is a series of look and walk actions during an agent's lifetime to reach its maximum fitness. And finally, an evolutionary strategy, which uses a genetic algorithm to evolve this developmental strategy from set starting locations. First up, we're going to discuss the NK fitness landscape. The NK fitness landscape is a fitness landscape with 2 parameters, N and K. N is the size of the landscape, or also the length of the encoding of a genotype. K is the ruggedness of the landscape. This is how many factors the fitness of each gene locus depends on. Moving forward, we're going to use location as an analogy for the genotype to avoid confusion and make visualization a lot easier. Next up, we're going to discuss the developmental strategy of an agent. At each step in an agent's developmental lifespan, it can take 2 possible actions: look and walk. During a look action, it will gather information about its surrounding landscape by randomly sampling a location of the landscape exactly 1 bit away from itself and adding it to a list of observed locations. During a walk action, it will move to the observed location with the highest fitness, or if there are no observed locations, it will move to a random location 1 bit away from its current location. After each walk action, the set of observed locations is reset. This sequence of actions an agent takes in its lifetime will be predetermined.
Here's an example of a developmental strategy. If you look at the figure on the right, you can see that it's broken into 4 sections, A, B, C, and D. Each of these sections represent the same agent going through the same developmental cycle at different stages. If we look more closely at section A, we can see that the agent here is on a n equals 3 landscape, with each of these circles or nodes representing different locations, and the black node representing the location that the agent is currently at. The numbers or question marks within the nodes represent the fitness values at that location. For example, the agent is currently at a fitness value of 3. If there's a question mark in the node, this means that the agent does not yet know the fitness value at that location. So we start here with the genome, and the agent first takes a walk action indicated by the W. Because it does not know any fitness locations or fitness values yet, the agent takes a random walk to a random adjacent location, this time to the right. Next, the agent is instructed to take a look action. Because the agent does not know the fitness at any location, it takes a look at a random adjacent location and finds that the one below it has a fitness value of 4. Then the agent is instructed to take another look action, here in blue. So this time it takes a look at a different random adjacent location. So this time it takes a look up and to the right and finds that the fitness value at that location is 8. Now the agent takes another walk action. This time, because it knows the fitness value at these locations, it walks to the location with the highest fitness value, this time being up and to the right towards 8. As you can see, the figure on the left represents what's happening on the figure on the right in 3D. As you can see, the agent first goes down in fitness, indicated here, and then is able to go back up in fitness, indicated here, with the colors matching between the figures. This shows how through our different actions, an agent can escape a local optima.
All right, so for the experimental setup, there's a few different things that we'll be keeping the same across each experiment, so we just wanted to say these now to be as clear as possible. So first off, the starting location for agents do not change on the landscape, so each experiment evolves different strategies to maximize fitness starting from the same point. So each strategy always starts at the same point on the landscape to allow for a more consistent analysis. Secondly, all landscapes are n equals 15. K-values change across the different experiments, so those will be specified, but every landscape is n equals 15. And finally, all strategies are constrained to have exactly 20% walk steps and 80% look steps, depending on strategy length. This allows for easier comparison between our strategies, and this 20 to 80 ratio was chosen as a result of the naturally evolving ratios when these strategies are unconstrained. So in our first experiment, we looked at the relative performance of different types of strategies across different landscapes. So these are the average final fitness values after the strategy is executed on 500 different landscapes, with k-values ranging from 0 to 14 on our x-axis, and then the final fitness is on the y-axis. These strategy lengths are 200 steps, so 40 walk steps are present in each strategy. And what we can end up seeing is that the random walk strategy always does poorly, as it never makes any intelligent decisions. But then between our 3 more sophisticated strategies, being the steepest hill climber, the alternating strategy, and then our evolve strategy, we see that at higher difficulty values, the alternating strategy and the evolve strategy tend to do better than the steepest hill climber. And this is a result of the fact that combining steepest hill climb and random walk steps is very important when there's several local optima present at high K values. For our second set of experiments, we looked at the distribution of the look steps in the evolved strategies at some select K values. Here we have K values 0, 2, 4, and 6 represented. On the x-axis, we have the walk numbers. So these strategies were 50 steps long with 10 walks in them each. And then on the y-axis, we have the number of looks before each walk to make the whole strategy. So what we can see from this is we can see that at lower k-valued landscapes, such as k equals 0, we end up having very high numbers of looks at later walk steps as it tries to find the peak of the landscape, since k equals 0 landscapes are quite simple. Whereas when we reach more difficult landscapes, such as k equals 4 and k equals 6, we see the look steps be more distributed through life, as it's not as beneficial to use a purely exploitative strategy even at later steps in life once the landscape becomes more difficult. Alright, so for our third experiment, we investigated the random walks that are present in our evolved strategies at different k values. So again, we have the walk number on the x-axis, but this time the y-axis is how frequently we can expect a random walk to be present at these steps. Again, a random walk is a walk step with no look steps before it, so a purely random decision is made even if it can hurt the fitness of the agent. So what we see is in a landscape, like a k=0 landscape, we almost never see random walks present as they are not beneficial and could decrease the fitness. However, once the landscape gets more difficult, we see more random walks present, and this trend continues. So at higher k values, we see more random walks present at later steps in life. This is a result of the need to escape local optima, as the only way to escape a local optima is to take a purely random walk step.
From the results, we develop and discuss three key insights on the relationship between development and evolution. First, random exploration and development is shown to be necessary to survive selection. It is interesting that evolution drives organisms to ignore environmental cues at times against the promise of a fitness ascent. But randomly exploring populations diverge across larger search space, avoiding convergence to local optima. This may help explain the emergence of developmental phenomena such as child rebellion, where evolution has selected for populations whose adolescents deviate from developmental trajectories fostered by their parents to achieve greater phenotypic and behavioral diversity. Second, evolution drives development to be time-sensitive, with non-uniform patterns of exploration and exploitation throughout the developmental period. Evolved strategies in various landscapes all consistently exhibit different degrees of exploitation and hence those of exploration at different points in development, especially in the early and terminal stages of development. The continuous spectrum from full exploration to full exploitation can also be thought of as different degrees of sensitivity in development. Similarly, sensitive periods in biological systems, such as in humans' early brain development, demonstrate time-sensitive development arising from evolution as observed in our model. Finally, we find that evolution selects for a more complex developmental strategy marked by multiple transitions between predominantly exploitative and exploratory phases as the fitness landscape becomes more complex. High-K landscapes exhibit early exploitation and middle exploration, whereas low-K landscapes exhibit the simpler early exploration stage. It is surprising that nature and this highly abstract model share this trend of increasing complexity in development. As an example of a low-K landscape, flatworms exhibit a simple exploitative development, changing their size based on the nutrition available. In contrast, the cognitive development of humans exhibit multiple distinct stages due to the uniquely high number of interdependent factors from social influences.
So for future work, we have a number of interesting directions. The first is this notion of a multi-tier look, the idea being that rather only being able to look one bit away in the current model, would be able to look 2 or more bits away. We could think of this as a way of being even more sensitive to the environment and could be used as a way to investigate the evolution of sensitive periods. Another possibility is, rather than starting from a single location and evolving developmental strategies from that location only, we could actually allow evolution to evolve starting locations in addition to the developmental strategy, this being a little bit more akin to a true Evo-Devo model. In addition to this, we could imagine having dynamic fitness landscapes, so rather than having a single landscape during the entire lifetime of an individual, it could actually shift over time, much like a seasonal change. This could show selection pressures for different types of the season. We could also look at longer-term changes over evolutionary time, such as climate change, and answer other questions about plasticity in general. Finally, we could look to encode other types of behaviors other than simple walk and looks. We could do actions that are actually reliant on previous actions, such as doing the same or opposite of before. Actually having a descend action to help escape local optima would actually be selected for. And in addition, we could actually look at lifetime trajectories and see, based on what has happened during an individual's lifetime, use that to guide their actions. So, that concludes our first future work. I'd like to thank everyone for watching our presentation. Special thanks to reviewers and people that helped us at different parts of the project. And we would love to hear your questions, comments, and suggestions. Thank you.
Great. Thanks to the authors for this video presentation. Please raise your questions. We have Edward Kim and several other of the authors in the audience. So maybe I start with one from myself. Your search strategy looks quite interesting, just also as an optimization strategy, and it reminds me a bit of simulated annealing with the twist that you decide how many steps you allow in the annealing procedure. Have you compared your strategy to any of the common search strategies for selected benchmark problems?
Jacob, you want to answer that question? I think that's more fitting for you.
Yeah, we haven't exactly looked at comparing our search strategy to existing techniques like that, but that is a really interesting thought.
So I think, yeah, simulated annealing or maybe taboo search also has some shared characteristics with what you have presented. So could look into this direction. Are there any other questions in the audience? Just raise your hand or unmute your microphone or put it in the chat if you prefer. If that is not the case, I would suggest we start with the next presentation, which is by Jonathan Bowen about two theories of responsiveness. So Jonathan, I think you can just share your screen whenever you're ready.
Get this camera set up. Okay. All right, how's this? Does everything look functional here? Good to go? All right. Okay, so hello. I'm going to talk about responsiveness. So here's what I'm going to do. I'm going to just introduce what responsiveness is. Then I'm going to describe two theories that have been given to explain what it is. And then I'll say a brief word of evaluation. So the notion of stimulus and response are everywhere in psychology and the life sciences. So here's just a little response eye candy. This is a stentor responding to some irritant. It kind of shrinks back and then it swims away. So stimulus and response are experimental psychology's main independent and dependent variables, respectively. And also, you tend to see something like response to stimuli or responsiveness in list theories of life. So what is a stimulus and what is a response and what is responsiveness? So I claim that there have been two sort of main approaches to this, or two main theories. So I'll just describe them both now. So the first one I'm calling a functional pathway theory. And this is a very old diagram from Descartes. This has been the dominant causal theory of responsiveness since at least Descartes. And so this image is kind of quaint. We'd probably say some different things about what's going on up here in the head now, but at a very high level of abstraction, the underlying picture hasn't changed much, actually. So in particular, you always see recurring in psychological theories three components. So there are always sensors, there are central components, and there are effectors. And those are arranged in a feed-forward kind of fashion. So let's just say something about what each of these components are. So sensors are transducers. They transform energy at a sensory surface of the organism, and then they output to the central component. Let's talk about the effectors next. The effectors transform input from the central component into activity or behavior. And so what can we say about the central component? So it kind of has its work cut out for it. It has to take input from sensors and produce output for effectors and do something in between, some kind of integration. So this is where you might put the task of cognitive psychology, is to say, well, what goes on in the middle? And it's natural to talk about that in terms of information processing. And so people apply this scheme to humans, organisms at large, and of course, it's used in the design of robots. So what do stimulus, response, and responsiveness amount to on this picture? Well, a stimulus is just an application of energy to the sensor. It's pretty straightforward. Response is a little more complicated. So a response is an activity that's produced by the effectors where that activity is the culmination of activity that began with a stimulus. So it has to pass through the whole sort of causal pathway to count as a response. Now, finally, an organism or a machine is responsive when at least some of its activities are responses defined in that sense. Okay, so now to the second theory. I'm going to spend a little longer on this because it's a little bit less familiar. But this theory, I wouldn't say it was ever put together in the same place, but I'm drawing it from a bunch of loosely allied approaches in philosophy and in psychology and in biology. I've listed some of the sort of sources here. So the American pragmatist philosophers, the functional psychologists, people who call themselves organicists, and some humanistic psychologists. So all of these people have in common that responsiveness is something that specifically organisms do, and they do it as a whole, and it manifests in their conditions of living. And its deeper roots, you could say, are with Aristotle and Darwin. And that might sound, you normally don't put those two together, but I'll explain how that works. So let's start with Darwin's theory of natural selection. So this is how, this is how species originate. And for it to get going, you need a population of organisms that are surviving and that vary and that reproduce and pass on their traits. So what can we say in general about these organisms, the population that's being, operated on or that's evolving? Well, most theories that try to say something about what an organism is in general have this notion of metabolism. And it's hard to escape this kind of idea because organisms, they're complex, and that's kind of confusing because complex things tend to fall apart, but organisms, they grow. And if they're injured, they repair themselves. And that's kind of weird. So they do that by basically taking in sources of energy and assimilating them to themselves, and then sort of getting rid of the waste products of that. So here are just two common kind of ideas about minimal models of life. Here's Ganti's chemoton. So you see this circle here, that's the metabolic process. And here is a very simple diagram of an autopoietic machine, which is basically continuously creating itself. And that too involves a kind of metabolism. But if you look at both of these diagrams, you see that they sort of have requirements. They have these arrows going in. And now I'm going to say something that'll sound very obvious, but it's very theoretically important, and that's this. Organisms have needs. So if an organism has a metabolism, that implies that it has a need. So a need is just a set of objective conditions that are required to sustain the organism's vital processes. So I'm using the word vital here because both of its senses work. It's vital, meaning pertaining to life, pertaining to these metabolic processes that constitute living things. But it's also vital in that it's necessary. If the process is stopped, the organism dies. That constitutes not surviving. So if living is constituted by these vital processes, survival amounts to reliably meeting your needs in a certain kind of environment, despite obstacles and threats. So interestingly, just from those premises, you kind of get a ballooning set of needs because there are as many needs as there are ways to thwart these vital processes. So for instance, does the organism have a metabolism? Yes. Then it has physiological needs. Is the organism vulnerable to environmental threats? Yes. Well, then it has safety needs. There are conditions that are safe and there are conditions that are dangerous. Does it depend on a social form of life? Is this some kind of social organism, like a bee or an ant or something? Well, then it has social needs. It needs to be integrated into the community that it depends on to continue living. So survival consists in meeting needs, and evolution is going to select for organisms whose activity accomplishes this. So what does that look like? How do you diagram that? Well, here, to answer that question, you have to ask something about the overall behavior pattern of the organism. So what does the overall behavior pattern of an organism whose needs are reliably satisfied look like? Well, it looks like a kind of integration with all of the need satisfying conditions for that organism. So here I've got, so this little green circle, that's our organism. You've got food over here, it's got a kind of relationship with that. You've got a house, so that's shelter. You could think of that as providing some kind of safety needs. Of course, not all organisms have houses, but you get that I'm trying to depict something more abstract here. And then you've got, the other organisms. So here's a conspecific, and here's its community. Our organism is integrated into its community. So the whole pattern of life, you're going to see this organism going between various places, doing various things, all in ways that reliably lead to its needs being met. But these functional relationships, they don't come from free. They have to be established. They take time to establish. It's a developmental process to establish them, and you have to reestablish them when they break. So this is where responsiveness comes in on this picture. So here is a great term, red integration or read integration. So here's just a little dictionary definition. It's to restore to a state of wholeness, completeness, or unity. So this notion is used by Dewey when he's describing what stimulus and response really amount to. And stimulus and response on this picture are sort of phases of this process of basically forming an integration with something that an organism has not yet formed an integration with. So it tends to proceed in 4 phases or steps. So here's our organism with its sort of settled way of life. So let's just draw a little happy face there to kind of depict that. Well, then there's some kind of disturbance. Something happens, something changes in the organism's environment, a threat emerges, or an opportunity appears, or something that has been relied upon changes. Well, then the organism enters what Dewey calls the first phase of tensional activity. So the organism is not going to be doing the same things it was doing before it came into contact with this change, which it needs to integrate its activities with respect to. And so the state that the organism is put in, we would expect to see converge on being something apt to the kind of adaptation that needs to be formed to the threat or to the opportunity or to the change. So after that, you get something Dewey calls organismic search and exploration. So here, the whole organism's activity is kind of reconfigured so as to be selectively exploratory and to locate some relevant need satisfier or means of using and enjoying that need satisfier. So here, if he's found a dagger or something to protect him against this tiger. So now this final phase tends to be called consummatory activity. So this is either eating the food when you're hungry, neutralizing a threat, like in this case, I suppose, or maybe completing a job interview if the need is a social need for employment. So at the end of this process, the organism's enlarged its integration with its environment, and that includes altering its habits and the habitat that it's in. So what is a stimulus and a response on this view? Well, a stimulus is basically corresponds to the second step, this entering into this tensional state where the behavior changes of the organism. And a response is basically steps 3 and 4. it's the organismic search and exploration, and then the sort of consummatory activity, which restores integration with this new disturbance. So an organism is responsive on this view if it has a tendency to undergo this whole process of red integration. Now, I don't know if I have time for this, but so just to sum up, here are some differences between these two views. So the functional pathway theory of responsiveness takes it that responsiveness is about implementing a certain kind of abstract architecture. The vital integrative theory, on the other hand, really focuses on the relationships between the organism and its environment insofar as they meet its needs. Now, how do we understand the causal basis of responsiveness on these two views? Or how much do we understand it? Well, on the functional pathway theory, we sort of feel like we understand it in theory. But on the vital integrative theory, there's a lot of work to be done here. What is the causal basis for something which can build up these integrations with its environment? So there's a lot of good work to be done there. For the explanatory style of these two things, of these two theories, the functional pathway theory is clearly mechanistic, whereas the vital integrative theory, because it's always talking about the organism as a whole and its pattern of life activity as a whole, it's kind of a holistic approach, maybe an organicist approach. And finally, one more important point is that on the functional pathway theory, the causal processes are in the organism, whereas in the vital integrative theory, they sort of span the organism and its environment insofar as they are relied on for the life processes. So in conclusion, it's clear that this functional pathway theory has been more or less completely dominant in the cognitive sciences and in the life sciences, unless there's something I'm not thinking of. And on the other hand, this vital integrative theory, it's kind of been put aside. It kind of fell out of favor in the mid-20th century. It had a little bit of a heyday before then. But I think it has a lot of things to teach us with its focus on how the organism kind of, on its relationships with other things that it relies on. So I think there are a lot of resources to be recovered in these traditions, and hopefully it's of use. So thank you. I hope that was stimulating. And if you want to respond, now is your time.
Great, thank you, Jonathan. So it's a really interesting talk. There's a question from Vincent in the chat. Are there new predictions about organism behavior, evolution, etc., that this new theory makes that the old theory does not?
Good question. Off the top of my head, if you were to remove something that this organism relies on to meet its needs, you would see a kind of compensatory adjustment. You would see it find something else to fill the role, and you would see it establish that role. So for instance, if I woke up this morning and my fridge was gone, I have to find a new food source. I'd probably have to order Uber Eats or something as I get my fridge installed. But so here, you can make that prediction about my behavior before I've come into any kind of contact with the fridge. So that's one sort of class of predictions you could make off the top of my head. I think that works. I see a follow-up.
Yeah.
So should I take that or?
Please.
Okay. So can I interpret this as a stimulus, hunger, and response, behavior? Well, so we're trying to decide what stimulus and response amount to. If we're talking about this vital integrative theory, the stimulus would be it would be me entering this state of hunger, I suppose. I'm not sure if this is the best example, but the stimulus is when some change in the environment occurs and the organism enters into some kind of tensional or motivational state. So it's an intrinsically motivational theory. The stimulus is the formation of that initial motivation. The response is whatever you do to resolve that motivational state.
There's another question by Sharon Minsuk. It seems odd to ask which of these theories is better. That's like asking whether it's better to describe the operation of a car in terms of the engine and the gasoline and the drivetrain, or in terms of the driver and their needs to get to a destination. The two theories serve different purposes and neither is better.
Yeah, I'm going to agree with you. That's probably was not the best way to compare and contrast these views, or the best way for me to frame it. One interesting question worth asking is whether these theories are compatible or whether they're not. In the two cases you contrast, clearly it's compatible with the same vision of reality that gasoline, or the role that gasoline and drivetrain plays in the engine, and the fact that a person drives the vehicle, and where the vehicle is depends on where the person wants to go. So those are clearly compatible. Do these two schemes give us incompatible views of the organism and of the patterns that constitute stimulus and response? They might. These might be conflicting theories. But more work needs to be done to clarify that. I plan to explore that.
So to pick up on this line of questions, I wondered whether your vital response theory just refines or needs a refinement of the central processing units of the mechanistic theory, so that in order to be responsive, they cannot just be a black box, but at least a black box that has a certain behavior or meets certain requirements.
Yeah, so that's a good question. One thing that's sort of interesting is that on the second view, it's a lot more open to understanding the actual mechanisms of the organism as depending on outer objects. So they sort of pivot on things outside of themselves, and that's how they work. And a lot of these kinds of integrative, regulative control sort of processes, maybe they can be distributed throughout. First of all, they involve the whole organism. So, I mean, if we think about an activity like chasing, I'm not sure that's actually a good example. So the thing about, so the central component is a component mechanism in like a sort of input-output system, right? It's describing how some part of the organism is contributing to its whole pattern of activity. And so just to clarify, was the question whether the vital integrative theory could just incorporate the notion of a central component, like does it, or does it require it? Was that the question?
Or whether it refines the mechanistic theory by adding requirements to the central processing unit, like being able to be in different states of alertness or readiness.
Right. Okay. Yeah. So can you account for these bigger patterns of, entering into a motivational state and then doing things like altering your activity and searching for certain sorts of need satisfier? Is that something that you could give a theory in terms of the central component to account for? And yeah, I think you could try to. I think you would probably want to use notions of representation, representing the problem, representing certain value states, etc. And I think that probably is a very familiar approach to solving ecological problems of that kind, is to think about how can you represent the problem and how can you, represent the mechanism that instantiates it, or maybe a program that instantiates it. So that's that is one approach. You could take the patterns that the vital integrative theory describes and try to explain them using a mechanism like that. Could do that, and most people have been doing it, I think.
So your talk actually stimulated the audience a lot and led to a lot of responses, which are in the chat, and we couldn't address all of them. Maybe you all bring this discussion later into the Slack channel.
Yeah, totally. Yes, please send me your responses.
That brings us to the last talk of this session, which is by Anya Vostina, Catherine Skoselis, Alexander Lalayini, and Louis Zaman on the symbiosis and digital evolution of review and future directions.
Yes, awesome. So it's gonna be a little bit weird since the time slot is longer than I expected it to be. So I have a short recording that I will play, and then I have some pocket slides that I will switch to talk a little bit more, and we'll have plenty of time for discussion. So I will get the screen share going here. Oh, can I grab the screen share back?
Totally.
Jonathan, can you stop share your screen? No, I don't know how to. Just
wait, I can.
Oh, sorry about that.
Yeah, now everyone can override also.
And I'm not sure if I always forget to do right thing. It just keeps saying screen share is paused. That's weird. Do we have any idea how to make screen share not paused? Anyone? Anyone?
From our side, the settings look fine.
Yeah.
Interesting. I've not had this before. Resume share? No, that's not gonna work. Okay, let me move things across screens. Maybe that's the problem. No, no, okay. All right, I have one other theory of what's happening. Sorry, y'all. I have not run into this.
No worries.
Before. Okay, maybe this. Nope. Richard, I don't know, maybe something maybe what you just changed, because it just like every time I try to like resume screen share, it won't actually do it.
Try now.
Okay. Interesting.
Strange.
That's so weird.
We have to talk somewhere online where maybe Richard could play.
It is not uploaded anywhere, unfortunately. And I mean, I could switch to this. I mean, I could try sharing the slides. I don't know if that will share any better though, but we can try that.
Let's try it.
Nope, it's not gonna let me share that either.
Maybe try to, that has helped so far with most problems, is to leave and enter again.
Yep, will do. Will do. Good thing there's plenty of time.
Yeah.
All right, I'll be right back.
The computer science solution to every problem. Hopefully it works this time.
Yeah, these, what's called tech support people, they know what they're talking about, right? Now we have the normal settings here. Fingers crossed. Oh, this looks promising.
Yes. Okay. Yep. That turning it on and off again. That's always the trick.
As always.
The question is whether I shared sound though. I always forget to click those boxes. And I didn't. I assume you all want to hear the sound. Okay, here we go.
Hello, everyone. Thank you for watching our presentation. My name is Anya Vostnar, and today I'll be presenting on a review paper that I published with my co-authors back in the fall, focused on symbiosis in digital evolution. The paper is available and is at the link down in the lower corner if you're interested. So today I'll be talking quickly about what we mean by symbiosis, what we mean by this idea of digital evolution, and then finally how we can combine these and use digital evolution to study symbiosis. So first, symbiosis. What I'm talking about is a relationship where there are at least two individuals and they're from different species. And it is a net positive for at least one of them, but it doesn't have to be both. So it can be mutualistic where they both benefit. It can be parasitic where one benefits and the other is harmed. And it can be somewhere in the middle where one is harmed only a tiny bit and has some benefit or something like that. And then we're also talking about some sort of close and long-term relationship. So it's not just like a one-off interaction. They are kind of interacting long-term for a significant chunk of time. So of course that's fuzzy. And then this can be anywhere between obligate, where this is a necessary interaction for them to survive and or reproduce, or facultative, where it's not a necessary interaction, but is beneficial to one of them. So what do we mean by digital evolution? So if you've heard of AVITA, that is kind of one of the classic digital evolution systems that we're talking about. But it's generally this idea of we have an evolution of a population of individuals in a computer. So these are computer agents that have various traits. So they have to have a genome of some kind, and that genome has to be able to be replicated. So either they can copy it themselves or it can be kind of copied for them, kind of abstracted. But there needs to be some sort of replication with inheritance. And then there needs to be some sort of variation. So either a mutation during replication or crossover or some way for there to be variation in that population. And then differential reproduction, right? Some of them are able to reproduce, some of them are not able, and so they have different fitness and we get natural selection over time. So this is evolution by natural selection. So in AVITA, that kind of has classically looked like their genomes are sets of computer instructions, and the individual organisms act as little computers where they're able to execute these instructions, take in numbers and output numbers. Okay, so with that understanding, we were focused on how digital evolution has been and can be used to study symbiosis. So when we're thinking about how we combine these two things, we kind of have this general structure that most things fit into. So there has to be a world, that's just a digital evolution kind of standard that has organisms, and there's some sort of resource, in this case, CPU time. And then we have an individual host, and it might have some endosymbionts. In this case, we're picturing endosymbiosis, but there could also be cross-feeding between organisms, or ectosymbiosis, where the symbiont is like on the surface of its host. And both the host and the endosymbiont contain their own genomes and their own sort of process for metabolizing resources and executing their genomes. And what those details look like very much depend on the system, but some sort of process there, and then perhaps the endosymbiont is able to sort of interact with resources from the host. So digital evolution has been kind of a technique for about 50 years, since 1970, and symbiosis has been in kind of at the fringes of it that whole time. So it was found right away from the beginning, but wasn't always a kind of central focus. There's currently 3 active and open source systems by our count that support some sort of symbiosis in a digital evolution system. There's certainly also historic systems that don't quite work anymore, but could be revived. And there's a number of really cool example results from those systems. So I'll go over 3 of them today. One focused on coevolution with parasites, and that is impact on host complexity. One focused on the impact of spatial structure on the evolution of parasitism, and one focused on how symbionts of all varieties impact host population diversity. There's a lot of details that I'm skipping that are all available in the paper if you'd like those. Okay, so the first result here is this idea of coevolution with parasites increasing host complexity. So this is using the AVITA system. And in this system, organisms are able to do different logic tasks. So Avita is kind of a classic system for studying the complex traits. So for example, the not task only has this single logic gate that is needed versus the or task has 3. And so you can see that these are kind of objectively more complex and can be used to kind of understand how those traits might evolve and be impacted. So in this system, hosts are able to perform these different tasks to get resources, and then which tasks the hosts perform impacts which parasites are able to infect them. So parasites infect based on the host's resource uptake. So this is kind of mirroring the bacteriophage evolution or infection dynamics. And so this is work from Luis Simon, one of my co-authors on this paper. So in this system, Luis asked whether host parasite coevolution would result in increased complexity. Kind of 2 possibilities are that no, it wouldn't. It would just lead to this kind of continuous cycle where they go to the same attack and defense strategies, just kind of cycling around and not actually getting more complex. Or the alternative is that there is this arms race that leads to novel and therefore more complex strategies once all the simple strategies were exhausted. And so he was able to do a lot of really cool analysis because digital evolution allows for such fine-grained control. And in particular, he found that very clearly host complexity quickly evolved to be much higher when there were parasites than when there weren't, and even did some cool experiments where he removed them halfway through to see what effect that would have, and then also saw that complexity started to drop after that. And he was really able to dig down into why this was going on and found this kind of dynamic that he termed genetic memory, where the parasites were able to maintain the ability to infect these simpler hosts. And so we're kind of constantly pushing the host population towards more and more complex areas of the fitness kind of landscape in order to escape those parasites. And we're kind of smoothing out this fitness landscape, making those more complex tasks equally feasible. Okay, so switching gears, the next result that I want to discuss is in a different system and focused on what happens when evolution can occur between parasitism and mutualism. So in this system, simulation, hosts and symbionts are much simpler. They just have a number as their genome, but that number can go between -1 and 1. So very parasitic, antagonistic relationship or mutualistic, and can be anywhere along there. So with this system, we're able to look at what impact spatial structure would have on the evolution of either mutualism or parasitism or somewhere in between. And actually found that counter to a lot of what the cooperation literature finds, where spatial structure increases cooperation, in this system under certain environmental conditions. So this is looking at different vertical transmission rates, which is the likelihood that a symbiont gets passed to a host's offspring. At the intermediate ones, when there wasn't spatial structure, there was actually more mutualism or higher degree of mutualism than when there was spatial structure. So spatial structure actually selected for parasitism at these various intermediate sort of vertical transmission rates. So that has sort of interesting implications for when we would expect mutualism or parasitism to evolve. And then finally, a lot of these systems looked at the impact of these symbionts on host population diversity. So these were a lot of different systems that looked at these sorts of aspects. The Avita system that I already described, a system that had a cross-feeding relationship where organisms were able to output waste and other organisms were able to ingest that waste product. And a number of other systems. And so these found that both mutualistic and parasitic relationships led to increased host diversity, but for different reasons. So in the strictly parasitic, it was through the various kind of ecological and rapid evolutionary dynamics where the parasites target the most prevalent host, so it makes just diversity a kind of side effect. And then in the mutualistic relationships, there was often this sort of increase by the creation of new niches, right? There was more possibilities because there was waste product that someone else could ingest, or those sorts of reasons that would lead to increased host diversity. So same results, but through different mechanisms. Okay, So with that, gave you a very brief overview of this review paper. Like I said, there's a lot more details in the paper itself that I would love for you to read if you're interested. I talked to you about this idea of digital evolution, and it requires individual replication, variation, differential reproduction, and then how we can use that to study this phenomena of symbiosis. And the lots of really cool, new hypotheses and future directions that people have been using and can continue to use. There's a lot of exciting directions in terms of looking more into coevolution's impact on complexity and diversity and things like co-extinction, when we expect mutualism repair system to evolve in the first place, and when we expect symbiosis to evolve in the first place, and when we expect that to then lead to something like symbiogenesis, where the organisms are kind of seem to no longer be separate organisms. So we had support from a lot of different sources that are all on the screen there. And I would love to connect either on Twitter or via email and answer any questions. Thanks so much.
Excellent. Thank you, Anya.
Yeah, as I said, we have a couple of other slides if people are interested in the many things we skipped, but it seems fine to jump to questions too.
At the moment, if you have a question, just put it in the chat and raise your hand. And otherwise, you can present, I think, more from the slides you have. We are back on time because of the difficulty starting us off. But of course, I don't want to shorten your presentation time because of that.
Yeah, no worries. Yeah, so the other things that I just wanted to mention. So the paper also includes like a pretty detailed history of digital evolution, of symbiosis in digital evolution. So we did a pretty deep dive on like the historical systems like Tierra and even some earlier systems and looked to see where we could find symbiosis. So if you're interested in the history of digital evolution, I think it's a pretty cool kind of lens on which to look at it. And we also have like a whole section on future directions. So there's lots of this has been kind of an understudied area. And so there's lots of open questions that we are excited about and think that people could look into. In particular, I think there's a lot of possibilities thinking about how symbiosis impacts open-ended evolution. Oh, is there a question? Should I stop?
So, oh, sorry.
You can finish talking first.
Yeah.
But when you're done,
yeah.
you can the question. Yeah.
Yeah, and so then the one other thing is we also had this whole list. So of these many kind of things that we think like that there's a lot of potential to look at digital evolution and symbiosis with. We then also made a list of like no current system supports all of these different features that would be necessary in order to answer those questions. And so we kind of have a to-do list for the community, for all of us to start making some pretty strong progress in this exciting area. That's it.
All right, so there's a question from Seth, who is interested to see how you use existing models being extended to address symbiogenesis.
Yeah, so like I said, so this is, we were reviewing a whole bunch of different models. So I think that there's potential with a number of the different existing systems. The main thing is, so first of all, the question is, how do you measure whether symbiogenesis has occurred at all, right? And that's actually one of the areas, so the question of how do you measure at all, I think is a place where digital evolution can be really powerful as well, since I talk to biologists a lot, and often I'm asking them questions that like they've never thought about, right? Like, how do you actually like measure whether this thing is happening in the system? Because they just have the one system and like it's obvious like whether something's happening or not. So when you're kind of dealing with things that are in a gray area that you don't know, that's when you kind of really dig into how do you actually measure this? And I think symbiogenesis is one of those, right? It's a question of at what level is selection occurring? It's always occurring at multiple levels. Is our definition of symbiogenesis that it's occurring most strongly at the level of the pair? There's already obligate symbiosis where like they can't actually exist without each other anymore. Does that automatically mean that it is symbiogenesis? So I think that it's first we have to figure out those questions. And I think a lot of these systems like can already be somewhere where we're asking these questions just because they are a good example of like, well, they can already give up the ability to exist without each other. Is that what we mean here? So they wouldn't necessarily need to be extended in order to dig into those questions.
Great, thank you, Anya. So I suggest to address further questions to Anya in Slack. We're right on time for the next talk, but first I would like to ask you all to briefly unmute your microphone and just thank the previous speakers for their presentations. So. And I hand over the chair to Martin Biel, who should be in the room as well.
Yeah, can you hear me? Yeah, okay. Okay, so yeah, we go to the next speaker immediately, I guess, to save time. And this is Eduardo Izquierdo, who I've seen in the, who should be here. There he is. Hi, Eduardo.
Hey.
And he's gonna talk about some artificial neuroscience, maybe, I think.
Yeah. Hey, thank you. Can you hear me well?
Yep.
All right, great. So hi, everyone. Happy to be here. I'm going to be telling you about work that we've been doing in my lab to better understand what all of the different analyses of time series data from neural activity can tell us about the underlying operation of these behavioral circuits. This all work in collaboration with a former graduate student, Madhavan Kandadai. Right, so I'll start with the broader motivation for this work. I think we're living in really exciting times in terms of getting to study neural activity in relation to behavior. There's been incredible experimental advances that are allowing us to gather neural activity data from a wide range of living organisms at a faster pace than ever before and at a higher spatial and time resolution. And importantly, we're beginning to have access to whole brains at the cellular level. In some instances, even we can have labeled data for each neuron, so we know what neural activity corresponds to what neuron. And perhaps most importantly, and this is most recently, we're beginning to be able to do this in freely moving organisms. So that's huge. And so the goal of that push for data has been to understand the neural basis of behavior, of course. And so the question that we're asking is, how can we analyze that rich, complex time series of that neural activity to understand what the brain is doing and how it's generating behavior? And we don't know the answer to that question exactly yet. And it is becoming increasingly clear that computational models are going to be playing an important role in figuring that out. So there's been some work in this area using neural network models, but most of this has been disembodied, and they have tended to be more focused on the majority of on the nervous system itself, often focus on the statistical properties of the network and how signals propagate in the brain. However, by focusing on brains in a petri dish alone, it's not clear how much those studies tell us about the functioning of the nervous system as it relates to the complete living organism, how it generates behavior. And of particular interest, given how living organisms operate, is how these different neural activities relate to the different behaviors in different environments and different contexts that the nervous system is in. So the broad goal of this work is to propose the development of computational models of complete brain-body environment systems that can perform a variety of different cognitively interesting behaviors. That will be this blob here. And then to use all of the different kinds of analysis methods that people employ for living organisms, including particularly network neuroscience tools, dynamical systems tools, information theory tools, lenses on these systems, and then actually compare it to the underlying understanding of how these systems produce behavior. And that might be lesions to the nervous system or manipulations to the body or environment, informational lesions, psychophysic manipulations, and manipulations to the state of the system of one kind or another. And I think the idea would be that one would use this framework for determining the usefulness of certain kinds of tools or the lack of usefulness of some of them, most importantly, for building intuition around what it is that these tools are telling us about the operation of the circuit. I think that might be the most important one, and ultimately to build new tools or variations of the tools. So that's a really broad motivation. More specifically, I'm going to be working on perhaps the most popular network neuroscience tool, functional connectivity, as it relates to informational lesions. So the question more specifically for this ALife contribution is, what can these network neuroscience tools, and specifically functional connectivity, tell us about the behavioral functionality of a multifunctional system as described by informational lesions? The approach that we're going to take is we're going to evolve a multifunctional brain-body environment system. Then we're going to pick one to analyze this neural activity time series using those methods. And then finally, we're going to compare the insights gained from those methods to what we gain from an informational lesion characterization of the circuit. We're going to do that first for one, and then we're going to try to expand it for an ensemble of successful circuits. All of this work follows a computational neuroethology approach, picking a nervous system, embodying it, training it in some ways, in this case, for it to be able to do many cognitively interesting tasks or some cognitively interesting tasks. And then most of the work is on the analysis of the most successful of the agents in that population, again, through different lenses. So more specifically, with respect to the analysis, the idea is the two lenses that we're going to be using are, the first lens is we take that brain or that organism and we take it through the blue task, say, and we record its neural activity and calculate its functional connectivity, and that creates some sort of estimated functional connectivity map of the circuit. And then we take it through the second task, say this magenta task, some other cognitive task, record their neural activity there, the functional activity, and then the ultimate map of the circuit. And then we do the same, but now in a way that is not possible for most living organisms, where we take the organism through the same set of tasks, but we repeat the process as we informationally lesion either two ways, the pairwise interactions of two of the neurons, or one way, so individually for each direction. And we use the performance of the agent after lesioning to create a map of, in this case, not the correlations between the neural activity, but of the impact that a lesion, an informational lesion, has to the performance of that task. And so that creates a similar map that gives us, in some ways, what we'll imagine to be a closer to the ground truth of the functional connectivity of that circuit. And the idea is that we'll be comparing these two. We'll be using this continuous time recurrent neural networks. I won't go into the detail. And we'll be using agents from Randy Beer's visually guided, minimally cognitive agents. We'll pick two of these tasks, but again, of course, this can be expanded. So more specifically, we'll be using an agent that has 15 sensory neurons, 7 interneurons, and 2 motor neurons. The 7 interneurons are fully interconnected. So the agent can visualize objects as they fall from the sky onto it, and the agent can move left and right using those motor neurons. And we'll be doing 2 tasks. One will be an object's size categorization, where objects of different sizes will be falling from the sky. The agent has to catch the small ones and avoid the large ones. And the second task will be a perceiving affordances task, where 2 different shaped objects, in this case, rectangle type objects, will be again falling. And the agents have to gauge the gap between them and pass through, so try to center on the gap of the larger gap, so the opposite, and avoid the smaller gaps. And the tasks are sort of interestingly interconnected in some ways. All right, so the first step was to simply obtain samples of this. As far as we know, nobody has created or trained agents for multiple of these visually minimally cognitive interesting tasks. A regular evolution algorithm had no problem finding agents that could do multiple of these in the different tasks that we try, where again we tried those two. So we did 100 evolutionary runs and took the best agents at the end of the evolutionary run. And then we wanted to study only the most successful ones, or only the ones that were actually doing both tasks. So we did, the fitness is always limited, so we could only test some circle sizes, I think between 20 and 40, and some starting conditions. We then took those 100 best agents and we tested them with a smaller time step of integration and with many more trials. So during evolution, we tried 100 trials per evaluation. Here we tried 200,000 trials to really get at how well they were doing these tasks. And this looks something like this, where, for example, for one of the tasks, we have gaps that are small and gaps that are large. Or, for example, the starting difference between the gap and the agent, all of that, which is being varied. And then here in the color region, we have the final distance between the agent and the object. So this just means this solid colors on two sides means that ones where indeed the larger ones were passed through, so the distance is 0, and the ones that were smaller gaps were avoided. And so that's just an example of one circuit for one of the tasks, not even the two. But we did that for 100, and we focused our analysis on the very best one on both tasks. But we then do the analysis on, I think, the top 10 in this top right-hand circle. This is just an example of what the behavior looks like of this agent for a range of different starting conditions. So you have time on the x-axis and the offset of the agent and the falling gap or falling circles in the y-axis, and the colors here are simply denoting the binary condition of whether the gap in this case was smaller or larger in red, or the circle was smaller in yellow and larger in blue. So we're not keeping track of everything here, but for the most part, you can see that the agents are successfully being able to do this behavior in interesting ways and so on. And crucially, what we're going to be looking at is the time series of the neural activity. So this is just an example of that one best agent. Also, we're only going to be looking at the interneurons. We try looking at the sensory neurons and the motor neurons, but that gets complicated quickly. So we're just looking at the 7 interneurons, which are fully interconnected with their evolved weights and so on for the 2 tasks. And again, we can divide it again between the small and the large. All right, so if we have one of these agents with 7 neurons and a whole bunch of time series, we can do the thing that most network neuroscientists do, this functional connectivity analysis, which is just looking at the correlation between neural activity for a task. And we can do that for any pair of the neurons. So this will be the correlations for one of the tasks, where the white means that those 2 neurons were not, those pairs of neurons were not correlated, and the darker colors that they were correlated. Of course, we can ignore the diagonal and we can ignore half of the rectangle because it's a symmetric measure. And again, we can do that for the second task. So this is a perceiving affordances task, and this is the object size discrimination task. And of course, one thing to point out here is that then, even though the network is exactly the same, there's no plasticity. The functional connectivity maps for these 2 tasks are different. And then we can do the same, but now through the informational addition. So where we try to find what the performance of the agent is when we remove transmission of information between two neurons, between every pair of neurons. Again, we do this in a way that where it is symmetric. And so here, black means that removing that connection between these two neurons, say neuron 1 and neuron 4, decrease the performance of the agent all the way to 50%, which is as bad as it can get. And the white ones, or the lighter color ones, means that removing those connections did not make, informationally removing those connections did not make a big difference. And again, we can do that for one task and for the other task. Of course, quick things that I'll point out is for the same task, you can get huge discrepancies between the functional connectivity and the informational lesion, where in the one case, right, actually, I think I just realized I'm pointing out different things here. So let me go to I'm gonna move on to this kind of analysis. So it's hard to look at this map. So instead, we're gonna look at the relationship between the Pearson correlation and the two-way informational lesions, where we're gonna draw here each pair of these maps. And of course, what we see here is that we would have different regions. If we found pairs of neurons to be living up in this upper left-hand corner, then that would mean that there was no correlation and indeed no impact from removal. So that would be in some ways agreement. Same for this side here, high correlation and strong impact from removal, from informational lesion removal. And anything along that line is sort of agreement. And then on the opposite, we have the corners, which would be, this corner would be pairs of neurons that seem to be very highly correlated, but nevertheless result in no impact after they're removed, no impact to the behavior. And then on this case, the most counterintuitive of them all, which would be no correlation and nevertheless high impact from removal. Okay, so this
Eduardo,
is,
you've used 15 minutes.
Yes. I'm gonna be finishing this up here soon. So what we can see here from this data, these are all of the pairs that you see here. And this is for, the blue is for one task and the orange is for the other task. And what we can see is that for the most part, it doesn't seem like there's a great relationship between correlation, between the Pearson's correlation and the informational lesions. For the most part, there's an overestimation. There's a lot of neurons that say they are correlated, but actually have very little impact. And in some ways, there's some counterintuitive examples too, things that don't have correlation and nevertheless have an impact. We can repeat this instead of using Pearson's correlation, using mutual information, and we at least remove some of the counterintuitive examples, but there's still an overestimation. And then finally, we can do this using transfer entropy. In this case, the data is a little bit more subtle because it's non-symmetric, so we can do it for each pair in each direction. And we begin to see a little bit more of an alignment along the diagonal, but again, there's still a large set of overestimation. And then the final step is, that was just an example of walking through one agent. We can do this for the top 10 agents and more generally ask, how informative was any one of these measures? So Pearson's functional connectivity, mutual information functional connectivity, or transfer entropy functional connectivity, to how good were they at estimating the actual informational lesion loss by looking at simply the correlation. And the good thing is they're all negative, so they're along the right ballpark of estimation, but the Pearson's is the least correlated. Mutual information does a little bit better, and transfer entropy does just a little bit better. Okay, so just to summarize, I think I'm presenting here a framework for a way to begin to connect the type of analysis that people are doing on real neural activity to the type of things that we have access to with these computational models. The current insights are that at least these ones that we tried so far seem to be very poor correlators of estimators of what's happening inside the circuit. And where we're going next is, of course, beyond functional connectivity to other tools of analysis, including particularly edge functional connectivity that is very hot and being used right now. And of course, going beyond lesions to other kinds of analysis, dynamical systems analysis and state perturbation. All right, thank you. I know I'm up at the top all the time, but if this is something that you've been thinking about, then I'd be happy to chat about it over, on Slack or over email. Thank you.
Yeah, thank you, Eduardo. I think it's a very nice methodology. I'm a fan. Yeah, I don't know. We have maybe time for one short question, if anybody has one.
Eduardo,
yes,
can you extend this beyond node-wise? Can you look at like higher order clusters of nodes?
Yes, higher order is actually, yes, this was the most basic thing that we could do, but yes, the interesting stuff is ahead, and certainly what we want to do is move beyond pairwise. Thank you, Seth. Yes. Right.
Okay.
Thank you, fellows.
Thank you, Eduardo. We're gonna go to the next talk. It's by Catherine Scosillas. Maybe that was a wrong pronunciation, but I think I've seen her name somewhere.
Yep, it's pronounced Scotchless. So,
okay, it's coming up, but it's not there. Okay, now it's there. All right, go ahead, please.
So bear with me here. I am on rural internet, so thus the no streaming video here. But if you hear banging, there are guys doing stuff on the roof. So it's a great day for a conference all the way around. But I'm excited to talk to you about our project, The Evolution of Genetic Robustness for Cellular Cooperation in Early Multicellular Organisms. I completed this with my partners, Austin J. Ferguson, Clifford Boehm, Catherine Perry, Rosemary Adaji, and Charles Afria. As you likely know, the major evolutionary transition to multicellularity shifted the unit of selection from individual cells to multicellular organisms. Constituent cells must regulate their growth and cooperate for the benefit of the whole organism, even when behavior would have been adaptive were they fitting. This is a standard multi-selection problem where there is a low-level pressure to increase individual cells' fitness by reproducing as much as it can without regulating its growth, and a conflicting high-level pressure on cells to work with their kin by reproducing with restraint, because unregulated cell growth is usually maladaptive for multicellular organisms. It is known that there is a bias towards the high-level individual when the life of the low-level individual is tied to it. However, many factors influence this, and while the reproductive cooperation behavior is binary, the pressures that influence it are not. Mutations that stop cells from cooperating can lead to a variety of ailments, including physical deformities and cancer. Organisms therefore employ mechanisms to enforce cooperation, such as error correction, cellular policing, and genetic robustness. By looking at how large of a restraint buffer evolves, we can determine how strong the high-level pressure is. We measure a restraint buffer as the number of mutational hits needed to revert a multicellular organism to unrestrained behavior. We hypothesized that an organism fitness reduction due to spatial competition between cells can be sufficient to select for reproductive cooperation behavior in cells, and that this selective pressure would increase in larger organisms due to the greater need for cooperation. We built a simulation, Primordium, to study genetic robustness under a range of evolutionary conditions. We focused on early multicellular organisms with only one cell type, where cells must control their growth to avoid overwriting one another. In Primordium, cells replicate within a 2D multicellular organism, represented here by the blue square. Because every multicellular organism is this 2D grid, we can describe them by their height and width. For example, we would call this a size 3x3 organism. Each organism begins as a single cell near the middle of a square grid. Each cell has a replication timer plus uniformly distributed noise that starts when the cell is born. When the timer elapses, the cell randomly chooses an offspring position from its 8 neighboring positions. If a cell is restrained, it will cooperate with neighboring cells by not overwriting them with its daughter cells. If a cell is not restrained, it may cheat by overwriting its neighbors. As in nature, cells are subject to somatic mutations with each replication. Evolution between organisms favors reproductive cooperation because organisms reproduce when their grid is full, so restrained cells alone will result in faster reproduction for the organism. For example, here you can see a population of multicellular organisms with 2 dominant lines, pink and blue. The multicellular organism in the center is full. It selects a random position, and replicates into it, seeding the new cell with a sequestered germline, which also has a chance of mutation with each replication. However, unrestrained cells will outcompete restrained cells within an organism, as you can see by the unrestrained red cells overtaking the restrained white cells, hence the multi-level selection. We refer to the restraint value of an organism's germ cell as the multicellular organism's restraint buffer. As you can see, having more restrained germ cell allows the organism to accumulate more mutations before unrestrained somatic cells arise and take over. We found 3 key findings. First, we found that longer genomes reduce mutational pressure. To disentangle the effect of genome length on evolution at the population level from its effect on restraint decay at the cellular level, we built a control that artificially split the germ genome length from that of the somatic cell. This allowed us to test a range of genome lengths for the germ cells while always evaluating the organism using somatic cells with a length 400 genome. As you can see, we found that across every organism size, denoted in the gray labels, when genome length only affects mutation pressure at the population level, longer genomes consistently allow for larger restraint buffers to evolve. The restraint buffer for the smallest organisms level out once they can evolve to completely avoid unrestrained cells arising, but larger organisms monotonically increase restraint with genome length. The range of evolved restraint buffers also increases with genome length because all genome lengths have results that extend to the median of the genome, which is at a lower restraint value in larger genomes. Our second key finding was that selective pressure for restraint has diminishing returns. Here we examined the fitness benefit of 1 additional bit of restraint for different organism sizes and restraint levels. While increases in restraint are always beneficial, we observed a diminishing return in that benefit in practice. Small organisms see the greatest fitness boost just above the restraint threshold, after which the fitness advantage for additional restraint quickly diminishes. Organisms' buffer is high enough that unrestrained cells never appear during its lifetime. Larger organisms have a smaller initial spike, but given that they experience more cell divisions in their lifetime, this saturation point occurs at larger restraint buffer values. Thus, selective pressure persists to much higher restraint levels in larger organisms. Finally, our third key finding was that when all other factors are controlled for, larger organisms evolve greater restraint. When we removed evolutionary noise by simulating an infinite population, and we evened out selective pressures by simulating an infinitely long genome, we were finally able to see a monotonic increase between restraint and organism size. Here we show the evolved restraint buffer in the infinite population model, first for the 100-bit genome, and then for the infinite genome. Since this combination of controls changes the trend, we conclude that the downturn pattern must be a combination of decreasing selective pressure and increasing mutational pressure as restraint buffers increase. In nature, population sizes are limited, but are often much larger than the size 2000 populations that we tested. Similarly, while biological genomes are not infinite, they are typically significantly larger than modeled here. Thus, our system's infinite population and genome model highlights factors that could exist in nature. In this study, we have shown evidence that the pressure imbued by simple space management can select for genetic robustness to improve cooperation. Larger organisms have a fitness advantage if their genomes are more mutations away from an unrestrained state. From an initial transition to multicellularity, before pressures associated with developmental patterns existed, these dynamics would therefore be sufficient to create a selective pressure for increased levels of restraint on cellular replication. We did not accentuate the cost for unrestrained behavior because we wanted to explore dynamics that we believe reflect early multicellular organisms. However, once more complex aspects of multicellular organisms did evolve, such as developmental patterns, unrestrained cellular behavior, and especially unrestrained replication, are often fatal. As a result, the cells of multicellular organisms, even those with a body plan as simple as sponges, have evolved mechanisms to maintain appropriate numbers of cells within their tissues. Avoiding the turning point that we saw in Primordium required unrealistic controls, indicating that there must indeed be other factors in complex biological systems increasing the selective pressure for restraint. The dynamics we observed, however, may help explain the initial bootstrap as multicellular organisms evolved to be large enough for developmental processes to become beneficial. In future work, we look forward to building out more realism and complexity in Primordium, addressing additional issues relating to the transition to multicellularity. Thank you guys for your time.
Okay, thank you, Catherine. There is quite some time for questions. Maybe you also thought that you had less time than you actually had, because you had, there's still 10 minutes left. So if there's any questions for Catherine, please go ahead. Either put them in the chat, or you might also just unmute yourself and ask. And maybe while we're waiting, so one thing I didn't like, you mentioned at some point that you made some unrealistic assumptions, and I'm wondering whether one of those was this kind of replacing mechanism of one cell basically overwriting its neighboring cell, because that seems to be like in reality, if I think of some cell that's part of a multicellular organism, if it replicates, it would just push maybe it would maybe increase the volume of this kind of clump of cells, but it wouldn't need to overwrite any other cell. So
that's true, but the organism still has to support it with like resources. So if you think about a cancer tumor cell in your body, it is getting often stealing resources from the healthy cells. And in that way, you'll see damage to the healthy tissues around it. So that is definitely one simplification, but makes sense in context. What I was specifically referring to there was when we abstracted out to the infinite genome and infinite population models. So obviously, nobody has infinite DNA and nobody has an infinite population size. We needed to do that for our default 100-bit genome, which is on the opposite end, unrealistically small. So switching that to infinite meant that it was actually closer to reality. And similarly, like populations, our population size was 2,000, and switching that to a statistics model so that it could be an infinite population size let us run in a less than several months long simulation the ability to look at really large populations where you'd actually see these dynamics play out, whereas at the size 2,000, you had the influence of that really small population size. So I see a question from Francisco in the chat. The cells, they do not talk to each other. They do not look around. They do not gain resources. Everything that you saw in the presentation is exactly what they do. These were supposed to be simulating the most rudimentary cells that could be, kind of thing, because we were specifically wanting to look at that initial point transition to multicellularity. So there's no other influences there.
There's also another question now in the chat.
Yep, reading.
Okay.
So that's a really cool question, Anya. There's a whole bunch of work on simulations for cancer tumor evolution modeling. I know like Emily Dolson has worked on that, who's at ALife. And what little I know about it is that it's really useful for predicting the ways in which things will evolve so that they can kind of head them off with treatment, because often that's a problem in cancer treatment where the cancer evolves to become resistant to whatever chemo or treatment they're doing. But I'm not sure how our work specifically would play into that. I think to have real world applications, we would need to do future work to build in a lot more realism to some of the ways that these dynamics are influenced by things like circulatory systems and like body patterns and stuff like that.
Okay. Yeah. Yeah, okay. I agree with your answer to my question. I think it's a good point that like I can understand that one cell would take away some energy from another. So
Anya, are we planning to do it? I am planning to cure cancer during my PhD. I do have like 2 years left, so I think we have time.
No problem.
No, I don't know. That remains to be seen on if we will actually add to Primordium or start on another system here. Our lab is working right now on extensions to Avita, and I think that's probably where my next project is going to lie, is working with the new and improved Avita system.
I see. So Primordium is not the next version of Avita, I guess.
Primordium, its own little baby.
Okay. I see. Like probably much less ambitious than.
Yes, it is much, much smaller, custom made for this specific experimental setup program. We actually started with Avita, and it was just so much more complex than we actually needed to answer the questions we were asking, that we realized about a semester in that we were making life so much harder than it needed to be, and spending so much more time running simulations than we needed to, when you could really boil things down much more simply, which is how we ended up spinning up Primordium instead.
I see. Yeah. So is Primordium, is it still available online?
It is. It's on GitHub. If you go to the paper, it has access to the supplemental materials and the GitHub repo with everything if you want to play with it.
Okay, cool. Any other questions? There's a comment, I guess.
Charles with the hardball question. Advisors aren't supposed to do that. How a little change would be more complex with more organisms. We do see in nature the pattern, that trend where you see more genetic robustness with increases in size. But we also see a whole bunch of other ways in which organisms account for and counteract unrestrained growth. So like cellular policing and other things like that. And I think if we add in more complexity to the system, then we will see more forms like that being taken into account, because there's many ways in which, other than genetic robustness, organisms can account for cell overgrowth and competition. And I think you'd also start having, at least from my experience with that first semester of Avita, you'd start having more trouble piecing apart which factors of the environment and the setup are influencing which dynamics. But it could be really interesting if you could pull it apart.
Yeah, that sounds like a project. I mean, when you're done curing cancer, you can do that, I guess.
Lies, all lies. Thanks, Charles.
Yeah. So if there's no other questions, then maybe we can start a little bit early with the next talk, if that's okay. I mean, are we on the only track? Probably. Maybe it's not too bad.
Yeah, I think we are.
Yeah. Then maybe we can justify starting a little bit early. People won't be too mad. Okay, so the next talk is by Moyul Islam. Again, I don't really know if I pronounced this right. Are you here?
Hi, it's Moyul here. Hi, everyone.
Okay, so I can't see your slide yet.
I'll start sharing my screen.
Yep. Yep. Okay. Looks good. So take it away.
Okay, thank you so much. Hi, everyone. My name is Mohiul Islam. We are from Concordia University in Montreal. So the paper we are presenting here today is on a new programming language named String, which is designed for the evolution of ribozymes for a new computational protocell model. So the outline of the talk, I'll provide an introduction. I'll talk about the new protocell model. I'll talk about the language itself and how we can write ribozymes using the language. And I'll show you the results of the evolution of ribozymes. Then we'll conclude and also how we can extend the work for the future. So we all know that the cell is the unit of all known life forms, and all cells have three common subsystems. So we have the genome, which is the information coding for the proteins. We have the metabolic network, which contains all the chemical reactions that are catalyzed by the protein enzymes. And we have the membrane, which encapsulates all the activities of the cell, and which is also responsible for the exchange of material and information in and out of the environment. Sorry, I forgot to switch on my video. So if we go through the seminal work in artificial life, we will see that self-replicating digital organisms have been evolved in an information medium, and it has been done. It's a well-studied phenomenon. And we can also see study on having string molecules, automatons built using string molecules in artificial chemistry. And from a recent study by Das and Mitra, we can see that a whole cell simulation has been done where protein clusters are densely connected and spatially localized, but it lacks genetic representation. So our aim is to create a computational protocell which is inspired by biological protocell, and it mimics the essential processes of a cellular life, but not copying all the details of cellular biology. As you know, it's a computational model, so we need infinite computational power to do all the details. And so we would also like to create something autonomous, which has the essential properties of life, such as self-maintenance and growth. It needs to self-reproduce, and also creating a diverse population which can eventually evolve. So the model itself consists of atoms and energy at the very elementary level. So the atoms are we restricted the atoms to characters of SNOPs, which are basically carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, which are the most abundant elements available in all natural cells. And in our model, atoms are mostly molecules atoms consist of molecules which are passive entities, while the ribozymes are the active entities. So in our model, ribozymes carry out all the active operations within the cell. A ribozyme is an RNA molecule. It's a string of nucleotides, and the nucleotides also translate into the atoms consisting of SNOPs, as mentioned earlier. So the RNA molecule folds into an active molecule, which is a computer program written in string. And it's capable of processing the passive molecules. So in order to reduce the complexity of the different types of ribozymes that are existing in natural cell, we took 5 pure classes of ribozymes, and we wanted to abstract that. So we have transformers, which are analogous to enzymes that catalyze various reactions in the metabolism. We have translators that transcribe a gene into an RNA molecule and folds into a program. We have transporters, which are embedded in the membrane, and they're responsible for passing atoms and molecules or information across it. And we have replicators, which copy the entire genome and so that it can be passed on to a daughter cell. So this is an abstraction of our model. We have as you can see, the membrane on the outer side, and the small rectangles are actually the ribozymes, or the string programs. And while the curves that you see floating around, these are the molecules. We can see an metabolism activity happening here, operated by 2 string programs. And this is the genome, where we can see the replicator and the translator. So this is sort of a visualization of our model. So our goal is to create a computational model that has the 3 subsystems of essential subsystems of a living cell, the membrane, genome, and metabolism. So, and to do that, we designed a new programming language called String, which is also robust and evolvable. So the way we ensured robustness is because every genetic, if we create a random genome using the codons of AUCG, any random genome can be translated to a program. So our translation process ensures that. And we'll talk more about translation later on. So let's talk more about the language itself. So string programs are just a sequence of symbols, and any gene can be integrated to a program. It's the basis of the simulation. It's defined by an instruction set, and it doesn't crash. Given it's from any random genome, even though the genome is mutated, the mutated program does not crash. That's how robustness is ensured. So this is the grammar of the language. So it starts with a set of operators. And so every instruction set starts with an operator and an operator. So these are the list of operators that we have right now. 16 operators, which does cutting, joining. So these operations are performed on the molecule itself, and the molecule is contained by the registers. And we have eight registers. So the numbers here represent the registers. So if you perform a cut operation on two separate registers or one register, it's basically going to cut that molecule depending on where the cursor is present. So registers can also contain patterns. So patterns are basically atoms, string of atoms, along with wildcards. So that's how it recognizes patterns. So if I specify something like, let's say, site I, so that means it will site I over here refers to the internal environment, which is the cytoplasm of the cell. So it would cite a molecule with a certain pattern and put it in the register. So that's an example instruction. So this is how the translation happens, and this is also how robustness is ensured by string programs. So, given any random genome, which starts with a promoter and a start codon and an end codon, the section in between is being translated to a string program. And the way it does is it keeps looking for encodings which it can match. And as long as the encoding is not being matched, we consider it as a junk. So in this way, we translate the codon into an operation, into an opcode, and then type and a value. And this is how the entire program is translated. So here in the tables, you can see the encoding process. So when we see a coding of this kind, it's translated to a cut operation, and so on and so forth. And you can see the other type of encoding as well. So how did we evolve, right? Why evolution of ribosomes? So our ultimate goal is, of course, to evolve the entire protocell and a population of cells, but we haven't reached there yet. So before reaching there, we wanted to write ribozymes, string programs, and be able to evolve those to see and to be able to have confidence in the idea that this is actually worth pursuing. So the way we evolved, as because string programs are like regular programs, so we evolved it using a simple genetic programming algorithm. So our algorithm is quite simple in terms of GP. So it has an initialization, it has a parent selection to tournament, and it has two different variation operators, crossover and mutation, and finally child selection and the loop iterates. So the mutation operators are nature inspired. You can do a point mutation, insertion, and deletion, and they're all operated on the codon itself. So mutations may or may not lead to a change in the program, as you can understand through the translation process. So we designed a nature-inspired crossover operation. So this is how horizontal gene transfer happens between a pathogenic bacteria and a non-pathogenic bacteria, where a section of their codon is being inserted into a non-pathogenic bacteria, which results in two progeny. So that's our crossover operation. So these are the list of parameters that we use for the evolution. And I'll get to the individual programs that we evolved. So we evolved a mass transporter, so with a population size of 1,000. And as you go towards the right columns, the size of the program becomes more complex. An information transporter is more complex, so the population was increased to 10,000. And then finally, the replicator translator are the most complex programs. So the significant factor is, so we applied mutation. So we were up to the transformer, we were able to apply evolve to the best fitness using 100% mutation. But for the replicator and the translator, we also added 20% of crossover operation as well. And as you can see, the initial gene size also increases. So with the size of 100, we were able to evolve a mass transporter, but the replicator and the translator required something of a size 500. So let me get to, so this is the result of the evolution. So for the simpler programs, mass transporter, information transporter, and the transformer, we were able to get the best fitness. But for the replicator and the translator, we were able to, for the best fitnesses, we were able to go up to 97%. And this was up to over 50 run of executions. So this is how our mass transporter looks like. So this is the register number. So as soon as so once we execute the program, if an input molecule exists of a pattern like this, it is being imported to the register and then moved to the inserted to the cell. So that's the site I is basically the internal site of the cell. And this is our evolve program, which is basically functionally the same thing. It functions the same way. And these are the list of test cases that we used. So we put these molecules in the cell and we execute the program, which basically provides the expected output is this. So that's how we designed the test cases for evolution. And so usually once we perform each of these tests on the execution of the program, we take the minimum edit distance and we calculate the total minimum edit distance from the actual input, so compared to the expected output and the actual output, and which basically results in the fitness. The value is also normalized to stay between 0 and 1. So these are the test cases for an information transporter. So the information transporter expects a molecule like this in the external environment. So as soon as this molecule is being sensed, then two internal molecules of this pattern are being added together. So a transformation occurs. So, and here you can see the ideal information transporter. So it senses a molecule of Schnapp's pattern, and then if it doesn't find it, it jumps to show, which is basically, it doesn't do anything. And otherwise, it will come inside and it will take two molecules of these patterns, put in the registers, and then join them together. So the evolved version, functionally, it does the same thing. And this is the transformer. Transformer basically takes three molecules of specific patterns and then joins them together. That's a simplest possible transformer that we could devise for the protocell model for now. Eventually, we have plans for an entire metabolic system, but yeah, this is for now. So as you can see, the program, it takes three separate patterns, puts them into these registers, and then simply joins them together. And this is also functionally equivalent. So the test cases for the replicator is further more complicated. So the replicator basically expects to see a certain pattern, which is the origin of replication, OR. So once it finds this pattern, it basically starts the copying process. And then it will copy to the end and then come back at the beginning and finish until it finds another origin of replication. So this is the expected molecule is basically, you can say, put back together again, reformatted basically. So TUA, the original replication appears before, and then the rest of the other molecules. And that's the actual output that we got from the evolved program. And these are molecules that we leave within the cytoplasm so that the ribozyme or the string program basically does use them for creating a copy of the initial genome. And over here, we also have a mapping between, so what we mean by AUCG. So AUCG are actually molecules within the protocell model. So the molecules are translated to atoms in this pattern for simplicity. And these are the test cases for the translator. The translator starts with the start codon and an end codon. So as soon as the translator program will find this codon, it will start making a copy. As you can see, AUG was the copy. And then as soon as it finds the end codon, it will stop copying. So that's the output. And this is the actual output that we received. And these are the molecules that we left so that the copying process can happen. So these are the actual programs. The ideal replicator looks like this, and the evolved one which could not reach to the ideal fitness, but went pretty close, up to 97%. And the translator over here on the right. So I know it's not much visible, but it's there in the paper, for detailed study. So in conclusion, excuse me. So string, we present. What do we present? We present a programming language. We can use use the language to write and evolve ribozymes in a new computational protocell model. We provide handwritten and functionally equivalent evolved string programs of 5 different categories of ribozymes. And we did the evolution using 3 separate mutation operators and a new crossover operator. And the programs that were evolved are within tolerable limits. So in the future, we intend to evolve an entire protocell simulation where all the individual components will be written in string. And then once the entire cell is functional, along with the 3 subsystems, we intend to evolve a population of it and to see a collective behavior. So, and with the current results, it provides evidence that such an effort is excuse me, worth pursuing. That's it. Thank you so much.
Okay, thank you, Mohir. Okay, you stopped on time, so we have 5 minutes for questions. So again, if you have any questions, put them in the chat or just unmute yourself. And if there's nothing at the moment, then I'm wondering, like you had this one picture where you showed like maybe a sketch of the protocell or something, and how I didn't quite get how this this looks pretty spatial to me, and I don't really understand how the spatial thing happens in your, like, how do you say something like which molecule sees which molecule, or I don't like this. I kind of can't really map this picture to how the program is actually going to run that's going to simulate the protocell. How is this working?
Yeah, so we don't have a spatial representation right now. So the cytoplasm is basically a list in Java or C++, right? So it contains the list of molecules, it contains a list of ribozymes. So this is a complete abstraction of it, and this is not how the actual programs are represented. But if we want to visualize it in this way, if we put, let's say, for each molecules or ribozymes, if we put an XY coordinate associated to them, so it would be possible to visualize it like this. But right now, there's no spatial representation. Because,
so you can also just randomly select 2 things that are supposed to interact if you wanted to.
Absolutely. That's how it is. I mean, so basically, the ribozyme will pick a pattern if it's looking for a pattern. It will pick a pattern of a molecule, perform the operations on it, and then emit those molecules back into the cytoplasm. It basically gets added to the list.
So okay, it's like some kind of, I don't know, well mixed.
Absolutely. Yes.
Okay.
Yes. Yeah. Exactly.
Okay. Yeah. There's a question, I think, in the chat by Mitsuyoshi. Can we see the specification of string? Is it published on the internet?
Yes. It's in the paper. I have over here, I have the grammar of string. I didn't put all the detail of the instruction sets. So it's in the paper. You can see the individual operations, how it happens. So what exactly does cut do, what emit does, and their detailed instruction sets. So basically they're manipulating registers. So the information is contained in the register, and the join operation performs a join or a jump. So it's very assembly-like. Yeah. So the source code for the interpreter is not published yet. So we intend to do some more work, like build the entire protocell, and then probably we'll publish the source code. But for now, we have the details of the instructions are there in the paper.
Okay.
I hope that answers.
Yeah, there's a link. Yeah, Anya posted a link. So I think, yeah, looks good. Okay, thank you very much for your talk.
Thank you, everyone.
And yeah, let's go to the next speaker then. Unless there's somebody else now. And that's Penelope Faulkner Rainford. Is she here? Yeah, there she is. Okay. Yeah, I can't see the slide yet, but maybe it's coming up. If you want to try sharing. Looks good. Okay, it's there. So go ahead.
So hi everyone, I'm Penny Faulkner Rainford from Lancaster University, and I'm here today to talk to you about something a little bit different. So this is going to be a talk about using some ideas from more ALife side of things and bio-inspired ideas to help improve work in genetic code improvement, where a lot of these ideas are quite rare and not used much at all. Or there's quite a narrow view of evolution in that area a lot of the time. And there are reasons for that, and we wanted to explore beyond that. And this work was a collaboration between myself and Barry Porter. So the context in which we were using this was emergent systems, which are hopefully something that would also be of interest to the ALife community, but I think more on that maybe in the future. So these are software systems made up of lots of small modules where you can which run continuously and are improved, optimized, and maintained by modifying the structure of the modules and by updating the modules themselves. So long term, it should be possible to move things in and out of the structure, and also take individual modules out, fix them up, and replace them with that. And that's where the genetic improvement comes in. So the idea here is, while the system is running live, we capture some inputs to it. We pass that, the original code, and in our case, we also synthesize some extra code, because these code units are often under 200 lines in length, and some synthesized data to speed up the process, so we can capture a very small amount of data from the actual system and expand its size for training purposes. We run a genetic improver, we get an improved candidate, and we pop it back into the system. Now, to make sure we're not having to do this too often, what we actually want is quite a few variants. So when we see an environment again, we can swap to the correct variant. Or when we see a new environment, we can use the most appropriate variant at the time while we're trying to improve it and get a more optimized variant. So the obvious thought that comes to most people's minds for how we do this with evolution is something along the lines of Darwin's finches, where you start from one point and you end up with 14 different specialists who are all very well optimized to their environment, except the fact that this took about 2 million years to happen and 14 different fitness functions. It also occurred on an island. So a very enclosed environment with very little competition. Now, if we're talking about a live software system, things are changing a lot, and there's a lot of competition. So if your software can't stack up, other software will come in and do the job in the meantime. Right, so being an island is not really what we're aiming for, and taking 2 million years, even in digital terms, is a long time to wait for these improvements. But actually, evolution does work a lot faster than that. When the environment changes, nature adapts very quickly, either by new organisms moving in and taking over the niche, or actually by using existing variation. So the great example of this is the peppered moth. So these were not two completely different organisms. This is one organism with two variants, which over the course of a few years, in two different cases, managed to adapt from an unpolluted environment to a polluted environment And then when the Clean Air Act came in, it actually managed to adapt backwards. And this was because it had variance in the population from the start, and was able to use lineage selection to increase the rate at which we had one type of moth and decrease the other one. And then when the environment changed, it just swapped back. So there are ways of doing this much quicker, and lineage selection is a very fast way of doing it. So we want to look at, does that work in the context of genetic coding? So we just fell off the desk. So starting from this existing system that already did our code synthesis and data synthesis, we started with a hash function. Very nice, very simple little hash function. So the idea here is, if you want to optimize your hash table and how you store your data, you need to optimize your hash function to better distribute your values. In this case, we had this ht length object, and that was limited to 100. So what we want to do is make sure that whatever value we're given in as the key, we put things into these 100 bins as evenly as possible so we can retrieve them as quickly as possible later. And in this case, we worked just with lists of words. It was a nice, easy option, but the words were gathered from news stories. So we'd get repeats of words, and we get varying lengths and things that match the true distribution of a language in terms of letters, etc. And we put it through, and we can get an improved candidate like this, where you've got lots of repeated lines which come in due to crossover, where those lines have been copied from another piece of code. We've got some synthesis here as well. Result equals result plus result here. Kind of synthesized code. And we've been working with this for a while now, so we've got a whole load of these. So what we actually did is we went and looked at some of our old data, and we found two lineages, one from a run trained on English and one from a run trained on Polish. And these were mostly chosen for how accessible their ancestors were, because we didn't save out every single individual in these older runs. So we found some runs where a lot of the ancestors of the extant final population were in our save outs. And we found one candidate in English and one from Polish, because we know that these train very differently in those older runs. And when we wanted to think about this in terms of how do we use those existing lineages, how do we take these two specialized lineages and use them for lineage selection, we considered, so do we just take our final best individuals, throw them into the GI as the start points, and see what happens? And the main thought there was they're specialists. They're very special, our final individuals. They're highly optimized to their language, as they're probably not going to respond well to a new language. So instead, we considered looking at their lowest common ancestors. As you can see from these lower trees, these are the individuals at the top here are the individuals from which all of the extant and final individuals descend. All of them descend from this one individual. They're about 50 or so generations back from the end of the run. And the hope was that that individual produced the entire extant population. So hopefully they've got a little bit of evolvability still to them and should be able to adapt better to a new environment than the final individuals. And we checked, and they were definitely different, and the hash functions work differently. In one case, we're doing a product of the key values, but with this odd reduction by the length of the key in there as well. I don't really want to write that out as a mathematical equation. It got confusing. Well, for the Polish individual, by the time it got to this point, was literally just throwing back the result of 2 times the length of the word. Apparently that was an efficient solution for the Polish input. So just to check that what we were doing was feasible and wasn't too insane, we ran all of this against those same languages again. So we took our lowest common ancestor in English, our lowest common ancestor in Polish, and a mixed population. So our population size was 30. So in the mixed population, you'd start with 15 English individuals and 15 Polish individuals. And we then trained this back against the same data they'd originally trained on. And as you can see, not as strongly for the English, though this is statistically significant. The mixed population trains as well as the English individuals, and the Polish trains worse. And in the case of the Polish and the mixed population train quite well, the English trains much worse. And just to really make sure that these things were behaving as one would expect, we actually looked into which lineages were dominant in the population, and we found that in the training against the English data, the English lineage became dominant very, very quickly, and against the Polish data, the same happened again. All good so far. So now we want to see what happens when you take these two lineages and put them in a new environment. All right, so what we wanted to see was if we put against a third language, in this case French, does it select the correct lineage to train as well as if we trained separately on both languages and then chose the best individual at the end? Because if it can do that, it's halved our computation time. We've started from two separate points, got to the same endpoint with only one run instead of two. It works. So we can see here that it turns out that our Polish trained stuff worked better against French than our English does. And we get very strong results from that. And likewise, we also get this very fast lineage selection. In fact, it's as far, the lineage selection against French data was as fast as it was against data these systems were already trained on. So there really is no loss to having the two start points rather than the singular start point. And what we hope in the long run is this opens up a lot of possibilities. So this was the initial work. Moving on, the first thing we want to look at is higher common ancestors. Because if you look in the paper, it's not very clear here, but we did actually test those best individuals as well against the French data, and they performed almost as well as the LCAs, and we were wrong, and there wasn't much sign that the LCAs were any more evolvable than those final best individuals. So we want to have a look and see if higher common ancestors would actually evolve better, and so we might get some better training. Because as you can see with this, most of our things were so specialized that they mostly got worse, and it was a case of how much worse they get rather than actually improving when they saw this data again. With that, we want to look at using some evolutionary analysis, because we want these variants to work with. We want to get together a good, and having an evolutionary measure to help us identify good variants could be very useful in the long run. These particular ones were all lineages which all came from that very first original start code for the hash function. We want us, and this is quite common in genetic improvement, is the idea is I'm trying to improve this piece of, so I will start with this piece of code. Given the emergent systems setup though, we want to know, what if I start with two completely different sets of code? What if I start with two completely different hash functions? Can we still get lineage selection? Can we still get useful evolution and crossover out of those? Or taking it to an even bigger extreme, if I started with two different sorting algorithms, could I actually then work with two different sorting algorithms, and as well as optimizing my sorting algorithm to my current environment, my current data inputs, can I also select which algorithm in general I should be using at the same time as optimizing it? We do want to do some work with improving the code synthesis. It's very basic at the minute. It doesn't do many functions. And we do want, with that, as well as this start code, we want to build up a whole collection of it. And we want to have a curated collection of starting variants and sim code for insert. And that's going to require some machine learning to maintain that collection. And eventually, one of the things we want to do is, since we have this idea of lineages, we want to actually start identifying where we have a split. So as we evolve, have we actually got two lineage? Have we now had a, I don't want to say speciation, it's a bit of a strong word, but have we had enough of a division that it's worth now calling these two lineages and then tracking those? And if we have interactive output, then as a coder, being able to look at those different lineages and go, actually, that lineage looks more promising. Let's upweight selection for that, downweight selection for the other one, and be able to get some human input into our systems. So that's a very ambitious set of things we want to do. Luckily, we recently got some funding to go ahead and look into all that. And as a quick advertisement on the end, we will soon be advertising for a 3-year postdoc and a PhD to look into this stuff more. And if anyone's interested, please contact myself or Barry Porter at Lancaster. And if anyone's got any questions, I'd be very happy to answer them.
Okay, thank you, Penny. We have 5 minutes. So if there are any questions, please go ahead. I can start with one maybe. So I think in your paper, you also mentioned evolvability. But in your talk, I didn't really see it a lot, but I guess that's kind of also an interesting question, is like which programs are the most evolvable ones?
It's a challenging thing to tell from the outset because a lot of it, there's been very little work on that because most of these systems have their own set of mutations, their own set of crossover, etc. And a sort of detailed analysis is very hard because of the huge size of the possibility space for mutations one can apply to any of these things. It's absolutely astronomical. So you can't really do a detailed assessment. But actually, one of my other papers, we have looked into the starts of how do we do more in terms of those sorts of assessments. We've started looking at trying to do something closer to phylogenetics, looking at all the data we have from past runs. I've got a paper that's, I think, just in preprint for that now. And like in the long run, it would be really interesting to see if we can start getting these evolvability measures, actually looking at being able to assign to code fragments how evolvable they are, how much potential they have for being inserted into other code, and alongside that, starting to actually assess for this piece of code, what is the best possible mutations to apply to access its potential.
Yeah, you gotta evolve the mutation operator. I'm saying this as if it was easy. I'm sorry. This is not easy. But there's a question in the chat by Wolfgang, I think. If you start from 2 different programs, could you comment on how you want to do recombination?
Right. So I think. So currently, when I say that we do crossover, that's a term we mostly use to keep other people in the GI community happy. What we actually do is what I would call horizontal gene transfer, because what we do is we take a token from one individual and copy it into another one. There's no two-way transfer, and it's not like half the code gets swapped or anything. And certainly, I think that's where we'd start with recombination. But we'd look at sizes of blocks that we want to move across a bit more. And I think things we want to look into is this idea of templates for insert code. And if we can get that working for how we synthesize code to insert, we'll start applying those templates to finding sensible structures in existing code to pull out an insert. That's the hope anyway.
Okay, and now I also got another question. So how do you, like when you, I don't have much knowledge about these systems. So how do you make sure that the code keeps compiling, which is something?
Yeah, it's a big challenge. So it's one of the things that limits our code synthesis at the minute. And right now, right now we're doing it mostly by strictly limiting a lot of our mutations. So we do a lot of scope checking that obviously has some overhead to it, which says, you know, you can't delete this declaration if it's used later. If it's not used later, you can delete it, go ahead, fine. But if it's used later, you can't delete it. You can't delete a loop if it has anything inside it, things like that. And modifications are done very strictly within limits of typing, etc., which is, again, limits a lot of what we do. The argument there is that while there's quite a lot of overhead from that, if you take that away, what you gain in speed because you're not doing the scope checking, you lose in terms of the actual speed of the evolution, because now half your population functionally dies instantly. It's got no fitness whatsoever. Half your population just doesn't exist in the first place. So it's a trade-off. You take somewhat the overhead of code checking your mutations in order to keep a functional population, but even then you sometimes get stuff that doesn't compile, and I think that's always a trade-off choice you've got to make with this.
Okay, so I guess this answers also a question I was maybe gonna ask Mohul from the talk before, because they came up with this whole programming language, and I was like, why do they come up with the whole programming language? But I guess they exactly wanted to make sure that their code always compiles. So I guess they took the other road in this respect. There's another question. I mean, like, basically, like, the time is kind of over, but there's like 20 minutes to the keynote. So maybe if anybody can leave if they want to, but if you want,
and it might also be that Sylvia might want to test with the keynote speaker if everything's working. So, at this point, I will stop the recording, and then once Sylvia starts talking, then I guess you guys have to.
Okay, we have to. All right, maybe.
Yeah, I said they have. Okay. Maybe take it on Slack.
Yeah, I'll answer that on one. I'll answer that one back.
All right, then thanks everybody.